Thanks, Katie. Our next speaker is Dr. Sid Chopra. He is a McKinsey Research Fellow and psychologist based at the University of Melbourne in Oregon, where his research focuses on multiscale approaches to understand the biological causes of psychiatric disorders and medications.
Hey, everyone. Thank you for organizing this, Maya. So my name is Sid. I'm going to be talking about multi-scale research, and there's obviously a lot of ways to come at this. There's temporal, there's spatial scales, and I'll kind of be focusing on spatial scales because that's kind of what I've been working around. And when trying to talk about scales, there's obviously a lot of different methods in terms of simple correlational methods all the way to biophysical models that link these scales, as well as theories of neuroscience and brain function that are interscale, that are actually trying to bridge these spatial scales. But what came up when doing this and being a psychologist, I want to address a feeling. And the feeling when you do this kind of work is that you're a jack of all, master of none when you're engaging this kind of work, right? Maybe that's even being a little bit too generous. And the solution to that, the cure to that, is obviously collaboration. It always has been collaboration. So what I did was I went back in the last kind of 12 months and found a couple of studies in the literature that required large collaborative teams who were experts in different aspects of these exact spatial scales or methods that hone in on these spatial scales to have some really nice insights into neuroscience. So the first study was by Shihan Zhang and Avram Holmes, and what they did was they got a molecular phenotype and used it to infer a cellular phenotype and then associated that with large-scale brain architecture. So what they did was used single-cell sequencing data to extract the transcriptomic or RNA signature of different cell types from about 6 different regions in the brain. And they applied that to the Allen Human Brain Atlas data, which has much better spatial resolution, but not single-cell resolution of where this RNA, which cell type this RNA belongs to, right? And using that, they could get cell type maps for about 27 different cell types, including GABAergic SNGC neuron density across the cortex, and they showed that it was associated with large-scale functional brain features from healthy samples, in this case, a gradient of functional connectivity. It was significantly correlated with that, kind of validating these functional signatures that we have a cellular basis. And they took it a step further, showing that you can actually predict the resting state network a parcel belongs to based on its transcriptomic cell type concentration. So this is a confusion matrix with true networks on the Y, predicted networks on the X. So you'd expect the diagonal to pop out if you were making good predictions. And in this case, it was generally there, showing this link between molecular phenotype to cellular to brain-wide architecture. The second study was almost unbelievable when I read it. This is one of the most interesting studies that's been done in a while. And this was by Brendan Ng and Jeremy Herskowitz lab. And this bridged almost every spatial scale that I have listed there, right? So what they had was 90 older adults that were imaged with MRI prior to them passing away, after which they did a post-mortem analysis in 2 brain regions, the frontal and temporal region, and quantified all sorts of molecular and subcellular phenotypes. And in here I'm showing both synaptic density as well as synaptic type. So they got a single subject level measure of these molecular phenotypes, and what they found was that this was associated with the functional connectivity ante-mortem before death, right? And this is just one small result from this paper, but they also had protein expression, a variety of interesting things. So the previous research was kind of working on group-level healthy maps and correlating them, which is interesting, right? But to get, I think, medical and actionable insight, this is the future, right? Single subject, intersubject variability. This sort of work is very difficult and requires collaboration across a lot of different teams, I think. And the final study is some of my own work. And sometimes it's hard because what Katie presented in terms of multimodal can be multi-scale as well, right? Because different modalities tap into different scales. So this is, I guess, a combination of those. And here we used a molecular phenotype to infer a subcellular phenotype and associate that with brain-wide architecture. So we used a PET tracer that binds to an SV2A protein, which is expressed on synaptic vesicles in the brain, and we use that to infer basically synaptic density. the number of synapses in a given brain region, and we did this in a patient population of people with schizophrenia. What we found was widespread synaptic reduction at the scale of the brain. And when we used that synaptic loss map to simulate how might this pathology emerge using diffusion data, right? Does this pathology start in one spot and spread to the rest of the brain? And we use large-scale axonal maps from diffusion imaging to actually infer that. So these are kind of 3 examples of what I think is the future, which is large-scale collaborations between people who specialize in methods that tap into different spatial scales. And to summarize, there's amazing studies like this appearing. It's not possible to be an expert in any modality, but open science and collaboration obviously are essential to this kind of work. Thanks.